Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. So thank you for joining us today. It is now my pleasure to introduce our speaker, William Rushton, process chromatography scientist from Bio-Rad Laboratories.
Thank you very much for that introduction. Today I'll be talking about a preparative purification of single-stranded oligonucleotides using anion exchange chromatography. I'm going to begin with a little introduction. Oligonucleotides play a fundamental role in gene regulation and are considered a promising novel class of biopharmaceuticals. The first oligonucleotide drug was approved by the FDA in 1998, which has led to a growing interest in the development of oligonucleotide-based therapeutics. Oligonucleotide-based drug platforms comprise many types of nucleic acids for specific gene modulations. These include antisense oligos, short interfering RNA, microRNA, and aptamers. My talk is going to be focusing on antisense oligonucleotides, or ASOs. ASOs represent an important therapeutic subclass and are short single-stranded oligos ranging from 19 to 22 nucleotides. They are manufactured by a cyclic solid phase synthesis and have a purity of between 75 and 90%. The product is mainly contaminated by shortmers, which is represented by N-X, longmers, N+X, and other structurally similar impurities. ASOs used in therapeutics need to be highly purified after this chemical synthesis. On the right, you'll see the product, which has 20 nucleotides, and the longmers would be, say, 21, 22, and the shortmers would be less than 20, 19, 18, and so forth. On this slide, we're showing a typical production and preparative workflow of creating the ASOs. So you have the automated solid phase synthesis, followed by a cleavage and deprotection, a HIC purification, followed by a detrotalization to remove DMT, an AEX purification, and then a UFDF. So the orthogonal two-step chromatography process after the synthesis would be the HIC purification and then the AEX purification. So since oligos are negatively charged at physiological pH and above, an anion exchange chromatography is used for the removal of length-based impurities, the shortmers and longmers that I referenced in my last slide. And the N-1 species are considered the most difficult to remove. So now I'm going to talk about the development work that was done with our AEX. So here the sample, we had a single-stranded oligonucleotide, a 20-mer and a 21-mer, shown there on the right. They were DMT off, desalted and lyophilized, and they have a phosphodiester backbone. The resin used was Nuvia HP-Q. It's a strong anion exchanger, and the column sizes used were 1, 5, and 25 ml, and the instrument used was an NGC-10, which is a medium pressure chromatography system with the NGC fraction collector. Now I'm going to talk a little bit about the Nuvia HP-Q Resin. So standard application for this is the purification of large biomolecules. It is a 50 micron particle with large pore sizes. It was actually optimized to allow large biomolecules to actually enter the pores. This was done by having an optimized surface extender length and density, as well as optimized surface area for easy accessibility, absorption, and binding. So this was why it was felt that for oligonucleotides that Nuvia HP-Q would be the perfect resin. Now, after discussing the method that we're going to use, I'm going to speak about our post-chromatography analysis strategy. We did some fraction purity screening using denaturing page with a 15% TBE urea gel. To the right, you'll see an example of the fraction screening. You have on the left the ladder showing the 21-mer and the 20-mer, and then as the fractions you can see where this separation occurs. Pooling is then done, and then further analysis of the fraction pool is done, again using the denaturing page gels and also an analytical AEX, which is a 1 mil enriched Q column. And you can see over on the right a chromatogram off the enrich showing the crude 20-mer with that N-1 side peak, and then in the orange is the purified where that side peak is no longer present. Next, I'm going to discuss the influence of the pH on the separation quality. So here we have a sample. It's a 1-to-1 mixture of the 20 and 21 mer run over a Nuvia HP-Q 4-site column, 5 mil column. So we have two different buffering systems. So the first one is a tris pH 9.5 eluted with 2 molar salt at the same pH. And then the second system is a 25 millimolar arginine NaOH at pH 11.7. And buffer B is the same with 2 molar salt. In both cases, the flow rate's 120 centimeters an hour. It's bound in a 2% B, and then the elution conditions are a 20 to 40% B in 20 CVs. The chromatograms on the right shows you that at pH 9.5 in the light blue, you're not seeing any separation between the two 20 and 21 mer, whereas at pH 11.7, you definitely see a separation between the 20 and the 21 mer. Now we're going to look at the role of the flow rate on separation quality. So again, we have the 1-to-1 mixture of 20 and 21 mer over the 5 mil Nuvia HP-Q column. For this experiment, we're only going to be looking at the pH 11.7, as the previous slide showed that 9.5 did not give you any real separation. We're going to evaluate flow rates of 120 and 240 centimeters an hour. And the same binding and elution conditions as before, binding in 2% B and eluting in 20 to 40% B over 20 CVs. And on the right, you can see in the blue is the 120 centimeters an hour, and the orange is 240 centimeters an hour. Both are showing some separation, but clearly the slower flow rate of 120 centimeters an hour gives a deeper separation between the 20 and the 21 mer. And this isn't completely unexpected, as usually if you want to increase your resolution, lowering the flow rate will get you there. So the final portion before we're going through the scale-up experiments is determining the binding capacity. Here we're going to be looking at 10% breakthrough. So we loaded a crude 20 mer sample at 0.2 mg per mil onto a 1 mil Nuvia HP-Q 4-site column. Again, using the same buffer system as previously done, flow rate 120 centimeters an hour. Binding is at 2% B. Dilution is 80% B because at this point, once we get that dynamic binding capacity, we're just knocking everything off the column. The results show that the DBC for the Nuvia HP-Q for this crude 20 mer is 20 mg per mil. Now on to the small scale purification of the 20 mer. So here we have 72 mgs of our 20 mer, which equals about 70% of the maximum DBC on a 5 mil Nuvia HP-Q 4-site column. Flow rate and sample binding are the same as the initial screening work. The elution conditions were slightly modified to 10 to 50% B over 60 CVs. The increased CVs were to account for the larger load. And also you'll note on the chromatogram on the right, the slope of the elution was changed during the run, as this showed some impact in the separation between the contaminants. So initially you had a little more broad slope at 10 to 23, then even broader from 23 to 35% B, and then a steep slope from 35 to 50% B. So fraction pooling after the page analysis shows the three areas listed in A, B, and C. So pool A contained the target and N-X contaminants. Pool B contained our purified target, and Pool C contained our target plus N plus X contaminants. Here you see the data analysis of the small scale purification. So the SDS page on the right shows the 21 and 21 mer ladder, and then you can see the crude material followed by Pool A, Pool B, and Pool C. A total of 56 mgs of the crude was loaded onto this column. Pool A contained 4.9 mgs of this, Pool B 51 mgs, and Pool C 0.5. But you can see very well clearly the N-X and the N plus X in pools A and C. The total yield for Pool B was 90% and had a purity of 97%. Next, we tried a small-scale spiking experiment to show the ability of the Nuvia HP-Q to separate the 20 and the 21 mer. So in this case, we spiked in 3 mgs of 20 mer into 45 mgs of 21 mer and ran it over the Nuvia HP-Q column. Flow rate and sample binding were left the same. The elution conditions were slightly modified to start at 2% B. This is to allow a better transition of connectivity after the sample application. And then you see zones A and B, and a detailed page analysis was done of zone A to show the separation between the 20 and the 21 mer. Here you see the page analysis of zone A, which was 18 mils. So keep in mind that the total elution was 170 mils. So this is about 11% of that total elution. As you can see, the first 7 mils contains the 20 mer, which in this spiking study would be considered the contaminant. In A2, which is also 7 mils, you see that transition where the 21 mer and the 20 mer are together. And then by A3, which is the last fraction, you see only 21 mer. This shows that the power of the HP-Q to resolve the 20 and 21 mer is pretty powerful and occurs very early in this process. The subpools were then generated and performed further analysis with analytical AEX. Here are the results of the page analytical AEX and the data analysis of this spiking experiment. So up top you see the SDS page runs of the fractions A1, A2, A3, and B, which is the main peak. The initial sample had 3 mgs of the 20 mer and 45 of the 21 mer. Pool A had 1.1 of the 20 mer, no 21 mer. Pool A2 had 2.1 of the 20 mer and 2 mgs of the 21 mer. A3 had no 20 mer, it was all removed by A2. And pool B, which is the main fraction of the 21 mer, had 41 mgs. So this resulted in a total yield of 91% and a purity of 97%. Now this is referring to the 21 mer, but it does show the ability of the HP-Q to separate the 20 mer from the 21 mer. Okay, the final experiment was to do a large-scale 20 mer purification. So here we loaded 400 mgs of the 20 mer onto a 25 mil Nuvia HP-Q column. The flow rate was slightly reduced to 100 centimeters an hour. Binding conditions left at 2% B. Elution conditions were the 2 to 50% B over 60 CVs that were done in the spiking experiments, and we kept the same slopes for as those runs. You'll see fractions A1, A2, B, and then C1 through C3. The fraction pooling after page analysis shows pool A1 is N-X contaminants. Pool A2 has target and N-X contaminants. Pool B contains only purified target. Pool C1 contains target and N+X contaminants. And pools C2 and C3 only contain N+X contaminants. Here shows the data analysis for that large-scale purification. On the right, you can see the SDS page runs with our pools A1, A2, B, C1, C2, and C3. And as you can see, A1 has just the N-X. Pool 2 has some of the target, which is the 20 mer, and N-X. Pool B appears to be pretty clearly just 20 mer. And then C1, C2, and C3, you can see the N+X's increasing as the fraction goes further out. So a total of 328 mgs of crude 20 mer was loaded. Pool A1 contained 0 20 mer. Pool A2 had 25 mgs. And Pool B contained 297. The overall yield was 90% and the purity was 97%. And this lines up very closely to the results that we're seeing in the smaller scale purifications. Finally, I'd like to just summarize the work that was done here. I think the data shows that Nuvia HP-Q Resin is suited for single-strand oligo purifications. Both the small and large-scale experiments showed high yield and purity. The use of the TBE urea gels enabled for quick purity checks after each chromatography run. And then finally, there's going to be some expansion of the purification experiments to looking at oligos having a phosphorothioate backbone. You can find application note 3290 on the Bio-Rad site, which contains all the full details of all this work. Additionally, I'd like to thank Anton Posch, who performed all the experiments for Bio-Rad, as well as Sharon Bola, who was the project manager for this process. So I'd like to end with just making you aware of some process chromatography resources for Bio-Rad. We have a global network of experts that can assist with your methods development. Additionally, we can provide process improvement assistance in optimizing your steps as you're doing your work. We provide continued support. We will assist with your scale-up, process transfer, and regulatory filings. I've been on site a number of times at CMOs for customers during column packings, so that is something that we do as well. If you need to see any of our application notes, they are available at bio-rad.com/library. You can request samples at bio-rad.com/resin sample, or you can contact your process specialist at process@bio-rad.com. Thank you very much for your time today, and I'd be happy to take any questions you might have.
Okay, great. As a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, would you be able to use the Nuvia HP-Q for oligos that are larger than 20 mers?
It's a great question, Alex. You could definitely use it for larger oligos. Really, with the large pore sizes of the Nuvia HP-Q, you could accommodate even larger oligos than 20 mers. Really, your limitation would be the ability of the HP-Q to be able to bind the oligos, and then it would just be optimizing your process to get the correct separation.
Okay, our next question is, can Nuvia HP-Q Resin be used for the purification of siRNA, miRNA, and aptamers?
That's a really good question, Alex. As some people may or may not know, oligonucleotide-based drug platforms comprise of many different types of nucleic acids. We discussed Antisense oligos here, but siRNA, miRNA, and aptamers also fall under this classification. So I feel pretty comfortable to say that Nuvia HP-Q would be able to purify those as well.
Okay, great. So it looks like we're running a bit short on time. So if we don't get to your question, we'll be passing them along to William, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, could you purify oligos in a single-step purification?
So that's not a one-size-fits-all question. It would be possible, depending on the that initial synthesis that occurs. If you have a pretty pure sample coming out of that, then you could possibly do that. But typically, you'll see the HIC done as the initial capture, and then the anion exchange column is used kind of for that final polishing, getting rid of any DMT on oligo species that might not have had the DMT removed. So it's possible, but usually you will need more than one step.
Okay, great. Well, thank you, William. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.